Good day, dear students. Welcome to the continuation of our discussion on leukopoiesis. This recorded discussion or lecture will focus on the development and maturation of our mononuclears, which include lymphocytes and monocytes. So for the mononuclears, I will start with the monocytes. So the monocytes, guys, are the largest of the circulating white blood cells in the body, and they play central roles in the initiation and resolution of the inflammatory response through phagocytosis and inflammatory cytokine release. Now, they also develop further and mature further to become macrophages in the tissues. You see in this figure the maturation and differentiation of the different progenitors and precursors of monocytes. Just like other white blood cells discussed previously, the monocytes developed from the common myeloid progenitor. For its earliest precursor, which is the monoblast, it differentiates further to become monocytes in the circulation and ultimately becoming macrophages in the different tissues and organs of the body. The development and maturation of monocytic progenitors and precursors occur in the bone marrow. The maturation of this white blood cell is similar to the process of maturation of the neutrophil, primarily because they share a common progenitor, and that is the common myeloid progenitor. For the major cytokine that controls the development and maturation of monocytes, we have the macrophage colony stimulating factor, or MCSF. The functions of the monocyte are divided into three. They play roles in innate immunity, they also play roles in adaptive immunity, and they also have housekeeping functions. So I will start my discussion on monocytic or monocyte development by discussing its first precursor, and that is the monoblast. The following are the characteristics of the monoblast. It has a size of 12 to 18 micrometers, a nucleus that appears to be round or oval, and sometimes it may also have an irregular shape. Inside the nucleus, 1 to 2 nucleoli may or may not be visible, and it will also have a fine chromatin pattern. Its cytoplasm appears to be light blue to gray, and it will have an NC ratio, or nucleus to cytoplasm ratio, of 4:1. For the reference intervals of the monoblast, it's not clearly defined or clearly identified as to how many are present or what percentage is present inside the bone marrow. And normally, or in normal patients, the monoblasts do not appear in the circulation. Inside the bone marrow, the monoblast develops into promonocyte. The promonocyte, therefore, is the second recognizable precursor of the monocyte. It has a size of 12 to 20 micrometers, an irregular folded nucleus that contains brain-like convolutions. Inside the nucleus, the nucleoli may not be visible. However, the nucleus of the promonocyte will have a fine to lacy chromatin pattern. The cytoplasm of the promonocyte is similar to that of the monoblast because it will still have a light blue to gray color. However, what's interesting about the promonocyte is the presence of fine azurophilic granules that have a burgundy color. Its NC ratio is 2 to 3:1, and inside the bone marrow, less than 1% of the nucleated cells is accounted for by the promonocytes. The promonocyte then develops into monocyte. Monocytes have a size of 12 to 20 micrometers. Their nucleus will not have a uniform size or shape, as they can be round, horseshoe-shaped, or kidney-shaped. The brain-like convolutions inside the nucleus may also be visible. Aside from this, the monocyte will also have a lacy chromatin pattern inside the nucleus. The cytoplasm of the monocyte will have a blue-gray color, and pseudopods may be seen. Many fine granules may also be visible, giving the cytoplasm a ground glass appearance. This cell appears both in the bone marrow and in the peripheral blood. They increase in percentage in the peripheral blood whenever someone is experiencing infections and or having autoimmune diseases. Monocytes still stay in the circulation for approximately 3 days before moving into the different tissues and organs according to the different patterns of their chemokine receptors. So once in the tissues and the organs, they then differentiate into macrophages. So we have macrophages in the liver, the lungs, the brain, skin, spleen, and all the other organs listed on this slide. So we have on this slide examples of macrophages present in select tissues and organs of the body. The photo or the image on the left shows a macrophage which is present inside the bone marrow, and the image on the right shows a macrophage present inside the bone. A general feature or general characteristic of macrophages is that they are generally larger than circulating monocytes. So that's it for monocyte development. Let me now move on to discussing how lymphocytes develop inside the bone marrow. So similar to monocytes, your lymphocytes are also important cells that play significant roles in immunity. Going back to this image, you will see that lymphocytes are quite unique in the sense that they are derived from a different progenitor. Unlike the granulocytes and the monocytes, which I previously mentioned, which are derived from the common myeloid progenitor, the lymphocyte is derived from the common lymphoid progenitor. The circulating lymphocytes are divided into 3 fractions or populations: the T cells or T lymphocytes, the B cells or B lymphocytes, and the NK cells or natural killer cells. The T cells are named as such because they develop and they mature in the thymus. They account for 52 to 81% of circulating lymphocytes, and they participate in cellular immunity. The next population of circulating lymphocytes are the B cells or the B lymphocytes, which account for 3 to 21% of the total lymphocytes in circulation. They develop in the bone marrow, and their main function is in producing antibodies, thus they are involved in humoral immunity. And lastly, we have the natural killer cells or the NK cells. They develop both in the bone marrow and in the thymus, and they participate in cellular immunity. The development of lymphocytes can either be antigen-dependent or independent. Antigen-dependent development of lymphocyte occurs in the bone marrow and thymus, or the primary lymphoid organs. Meanwhile, antigen-dependent development occurs in the secondary lymphoid organs such as the spleen, lymph nodes, tonsils, and the Peyer's patches in the intestines and other tissues in the body. B and T cell maturation initially occurs in the bone marrow and go through several maturation processes. The first precursor in the lymphocytic maturation is the lymphoblast. In normal individuals, lymphoblasts are only seen in the bone marrow. They have round to oval nucleus with more than one nucleoli present and a fine, evenly stained chromatin pattern. Also, lymphoblasts have scanty basophilic cytoplasm. You can see in this slide an image of an actual lymphoblast and its corresponding schematic diagram. Next to the lymphoblast is the prolymphocyte. Its nucleus may appear round and indented with one prominent nucleolus inside the nucleus. It will also have a slightly clumped chromatin pattern. Still, this precursor is not normally found in the circulation. The prolymphocyte then develops in the bone marrow to become a lymphocyte. Take note that B and T lymphocytes are morphologically indistinguishable from each other. The nucleus of lymphocytes is round to oval and sometimes may be indented. The scant or moderate cytoplasm will have a light blue color. Furthermore, lymphocytes are the only true agranular cell. However, when granules are prominent, the cell is called a large granular lymphocyte. So you see here on this slide a small lymphocyte on the upper left image, a large lymphocyte on the upper right image, and the granular lymphocyte on the lower image. And ultimately, when the B cells come in contact with an antigen in the secondary lymphoid organs, they undergo cell division and produce memory and effector cells. Effector B cells are plasma cells. Plasma cells are the cells responsible for the production of antibodies, which are important in humoral immunity. So for the characteristics of the plasma cell, they will have a round to oval eccentric nucleus with a coarse chromatin pattern and a deeply basophilic cytoplasm with a perinuclear halo known as HOF. This is the end of my discussion on the production and maturation of mononuclear cells. A separate lecture will be given about the specific functions and kinetics of the different white blood cells we just discussed. Thanks for watching this video and enjoy the rest of your day.